Thank you all for surviving day one, and now we are on day two. So I had a few questions coming from candidates from your class who were not exactly clear about what is the monoclonal immune response versus a polyclonal immune response, okay? And I think I might have dwelled a little too deep to explain things yesterday, so I'm going to try and simplify. If you have three different antigens / epitopes, right? That are recognized by these three different kind of B cells, as long as they are interacting with the antigen epitope of interest, these B cells will go through clonal expansion. Three different antigens, three different B cells responding to them. This is typically what you need to know as a monoclonal response. Now, there are three aspects you need to remember here or keep in mind. The amount of antigen will determine the amount of response. If you have more antigen, you will have more clonal expansion. That's number one. Second, there is an interaction over here with the antigen and the B cell receptor. If the affinity is higher, again the response will be more. So one aspect is antigen dose. That means a simple amount. The second aspect is the affinity of the antigen to bind to the antibody, right? And why I use the term antibody? Because the B cell receptor is actually the antibody, okay? If you have something binding weakly, okay, now depending on the affinity, if it is a very, very weak affinity response, you will have nothing taking place here. The B cell will come, try to recognize the antigen epitope, not recognize it, and move on. Okay? Let's say if the interaction over here is not as strong as the cognate epitope or the cognate antigen, yet it's not as weak as something it doesn't recognize completely. You know, let's say there's something in between. You have to understand biological systems don't come with an all or none response. Okay? It's a graded response that you can expect. So you can have a very high binding and a strong affinity or a weaker affinity or nothing below this, right? Now if this is a weak binding, it may or may not produce a response. Again, for something which has weak affinity, it needs to compensate with more dose. Do you understand? So if something has a weaker affinity, for it to produce an effect, you need to have a higher dose. Yeah?
But isn't this considered all or nothing because if there is a threshold?
Correct. So, but I'm just talking about the signaling part. I'm not talking about the effector part. You're right. Okay? Now, what happens is, each time your B cell engages an antigen, a signal goes inside the B cell. The signal can be weak, or you can have a strong signal going inside the B cell. Okay? And the strength of the signal is somewhat similar to what you would expect in an action potential. So if the signal remains weak and dies off, the cells don't get activated. But if it goes beyond the threshold, that's where the response comes in. Okay? Is this part clear to you till here? Interestingly enough, you may have a molecule that may come and bind to the B cell. Let's say this is the antigen, which is weak in itself. Okay? It's not really the cognate one, and it should not give rise to a strong activation here. But think about another molecule which is interacting and actually anchoring it together. So what will happen is, initially you will start off with a weak signal, and this weak signal will persist. Just because this molecule is holding the cell and the antigen together. Okay? And when this signal persists, this trickling over here that was weak keeps building up over time to the point that it starts going above the threshold. Okay? Now this is non-specific activation of a B cell. Okay? And if this molecule is engaging another B cell as well, and another one, all three will get activated with the same stimulus, with a non-specific antigen. Okay? And this is termed as polyclonal activation, where the stimulus that is being given over here is able to activate different clones or different types of B cells that would not normally respond to it. Okay? You understand this concept? Now these kind of antigens that are there are termed as superantigens, because they just go and non-specifically activate a group or cluster of B cells that would be there. They would do the same to T cells as well. Okay? So bear in mind again, monoclonal activation is with direct cognate peptide. Okay? And again, it depends on, and the cognate peptide, the one that activates it, it has a high binding affinity. Because it binds to it very well, it gives a push and the signal remains there for longer duration enough to get an activation spike. Do you understand what I mean now? Okay? But if your signal is weak, it would usually die off. Unless the amount is so much over here that the weak binding or weak affinity is compensated by the dose. Okay? And apart from this, if you have something that binds totally non-specifically, that's got no interaction to the B cell receptor itself, if it keeps binding and sustains for a long duration, okay, it will generate weak signals inside the cell. And if they keep persisting, they start adding up. They keep adding up to the point where they actually may get through the activation. Okay? And that's why you call it polyclonal, because the response of these cells would to become plasma cells, and all three of them, or many more of them, if you would want to be more precise, would start making antibodies that are targeting totally different things. Okay? So you will not actually get a targeted response to begin with. Okay? So is this clear to you? Make sense? Any more queries from last session? Yeah?
So in polyclonal expansion, the anchoring molecule has to be there, right?
Sorry? Yes. There should be an anchoring molecule that keeps it there, and the anchoring is on a site other than the actual antibody. It just makes sure that the peptide sequence that is not normally recognized. So normally, if you have a peptide sequence, and this is a B cell receptor with a B cell, it will come, engage, and because the affinity is weak, it will detach and move on. You know, these are molecular dynamics. So molecules come, attach, move away. If this is, let's say, the cognate one, it would kind of come and stick to it. And therefore, the amount of time it remains stuck to it would actually stimulate a signal inside. However, if this comes and something else comes and clamps them together, okay, this is a non-specific interaction, but this clamping molecule, the molecule that keeps them attached to each other, makes sure that the weak signals keep going through till it gets activated. Okay? Now, having said that, when we talk about the antigen governing the amount or the magnitude of the response, what does that mean? That means that this B cell receptor is in fact a mitogenic signal. What do you understand by that? Okay. Sabah al-Ward. Finally, at least we have some people waking up. So, what is a mitogenic signal? Yeah?
Mitogenic signal is a signal that induces mitosis.
Exactly. So, apart from all the things that we keep talking about, right? Activation, antibody generation. What's the first thing that happens over here? Clonal expansion, right? So before, you don't directly get a plasma cell. You don't directly get an antibody production. A B cell first has to multiply, expand clonally. So if you think about that, so B cell receptor is actually a receptor on the surface of the cell that regulates cellular expansion, right? Other signals, of course, will come in where they will start making antibodies and diverge. But the first step is that of inducing mitosis, multiplication, okay? So, in its very basic level, a BCR is a cell surface mitogenic receptor, okay? It just leads to clonal expansion. And after that, you have differentiation coming in, whether a group becomes memory or another group goes and becomes the plasma cells. Yeah.
Doctor, I don't really get the mitogenic signal. Like, what does it trigger?
Good. So a single cell primarily needs to divide. So, okay, fine, good you asked that question. So let's go back. Let's go a step back.
Can you say the question?
She couldn't quite get what a mitogenic signal actually means. Okay? So, let's say this is a pathogenic surface. Okay? And on the surface of the pathogen, you got different antigens available. Right? You have one particular B cell with a B cell receptor that will go and actually end up engaging the antigen. This is a very wrong drawing of what happens in terms of antigen-antibody, but this is just for simplicity, right? Because there's actually these tips over here that actually go and engage. But once this B cell is here, affinity, the high affinity that exists between the B cell receptor and the epitope of the antigen keeps the B cell engaged over there for a while. A signal goes inside the B cell to activate it. This B cell now goes into a stage of cell cycle and eventually divides into two daughter cells. These two daughter cells again carry the same B cell receptor. Now they cannot just keep dividing. It's a very regulated process. Like I said, the dose of the antigen governs the amount of the response. These cells will go back in the game and again try and engage the available antigen. And again come back, and now from 2 there will be 4. So it's a logarithmic division taking place. 4 will go back, 8, 16 and so on and so forth. Now the amount of antigen that is there determines, because at one point you will have too many B cells and not enough antigen over there. When you reach that stage, you will have a B cell that wants to engage to an antigen, but there's something called as steric hindrance, spatial hindrance, blockade, right? If I just want to run through here, To the back I can't. There's a table, there are students, there are chairs. There's no space for me to go to. They block me. Similarly, the B cells would be blocked by other B cells around it to go and bind to the antigen. Okay? So that becomes a limiting factor where the B cells cannot expand anymore. Okay? Does that make sense? Was that your question? Or did I go off in another tangent? Okay. Yeah?
The expansion,
Yeah.
it depends on the strength of the signal. But doesn't it also depend on how many epitopes we have?
So we are talking about one cell engaging one epitope. Okay? And that's it. So the amount of epitope is equivalent to the amount of antigen basically. We're talking one antigen, one epitope model right now. We're not taking into consideration multiple. Okay? So is this clear to you guys? Right. Finally, we can move on with today's stuff. Right? Now, eventually, your B cells, when they develop into either memory or plasma cells, they secrete antibodies, and the main cell that secretes the antibody is the activated B cell, which we call as the plasma cell. Now, the antibodies that you get that are actually present in the human body are of 5 different types: IgM, A, G, E, and D. Now, why have different antibodies when you are trying to give rise to or trying to maintain the immune system's functionality? Why do you think you need so many different antibodies?
Antibodies switching. Class switching.
No, class switching is the process that gives rise to them. My question is, why do you need them in the first place? Sorry?
Different pathogens.
Different body parts.
Different pathogens, different molecular structure. Any other ideas?
Different body parts.
Different body parts. Any other ideas still? Outcomes. You have different antibodies because the immune system is trying to have simply different outcomes. Okay? All of these antibodies are different in their size, shape, and a bit of character. And they're mainly different in their character. The typical antibody structure is made up of a heavy chain followed by a light chain. So you have one heavy chain which is engaged to one light chain, and this heavy chain is around 50 kilodalton in size. That's why you call it heavy. The light chain, of course, has to be less, and it's around 25 kilodaltons. And we discussed yesterday that that's how you measure molecular structures like proteins. And the heavy chain is linked by a disulfide bridge to a light chain. Now, the light chain here is in yellow and the heavy one in green. And just the mirror of this, another heavy chain with another light chain gives rise to the second half of the antibody. Just a duplicate structure, unlike in camels. Camels are single chained. So camels only have one heavy and one light chain. That's what makes them different from our antibodies. Although they are mammals as well. And they are found in this part of the world. So, yeah, not all my jokes are sarcastic. So you have two heavy chains which are linked to two light chains with a single disulfide bond, and the two heavy chains are linked to each other at an area which is called as the hinge region by actually two disulfide bonds here as well. Now, this structure is primarily called as an immunoglobulin, right? Why do you call it as an immunoglobulin? Globulin. What is globulin? Good. It's related to the immune system and it's a protein. But what's a globulin? What's typical for globulins? 3D what? Good. 3D structure. What is it more like? Do you know what are helices, beta pleated sheets? You studied this in chemistry? Good. So when you think about the light chain, it's got oppositely running beta pleated sheets, right? In all, there are around 7 of them, right? So 3 clustered together and another 4 clustered together. So when you look at the whole structure, when you make them, all these, and this is the light chain, by the way, okay? So when you look at one of this big rectangular structures, it is one cluster of oppositely running sheets, and similarly, this area is the other one, right? And in between them, they've got lots of van der Waals interactions that keep them together. Now, the heavy chains and the light chains that are present. So for the light chain, you've got 2 globular domains, and for the heavy chain, you may have 4 to 5, depending on the kind of the antibody. Now, apart from their structure, they have a variability in the sequences of amino acids that make them. So although this is one long chain, which is similarly depicted here, between different antibodies that are there of the same class, we're talking, let's say, we take an example of IgM or IgG. So within IgG and within IgM, these parts in blue are identical. There is no difference between them. Therefore, they're termed as the constant region. They are the same between the same class of the antibody. So if you take an IgG against influenza versus an IgG against polio versus another IgG against hepatitis B, among them in the heavy chain, these 3 globular domains will have no differences in between them. And similarly, on the light chain, okay, you have one area that is constant. The other parts, the most distal parts, the parts that actually are involved in engaging the antigen, they are the variable parts. Okay? So when you talk about, let's say you take a blood sample today from any one of you, and you pull out, you fraction and separate the IgG molecule, the constant parts will remain same across all of them. But the variable parts will be very, very different depending on the kind of pathogen or the antigen you were exposed to, and therefore you will have these parts that segregate one class from another class. So an IgG against hepatitis B, influenza, HIV, etc. Okay? And again, the more important part is that it's together the heavy chain variable part and the light chain variable part that both together form the antigen binding site. So the antigen recognition is not only due to a heavy or only due to a light chain. It's a combination of both that gives rise to this conformation where they actually go and bind to the particular antigen, right? Now, structurally, when they were trying to study proteins, one way to study proteins is to dehydrate them and crystallize them, because proteins, they can malfold, defold, pH changes, temperature changes can make so many different structures out of them. So this was the typical structure then that they got when they did a crystal structure of this particular antibody site. Now, the hinge region is actually, in the heavy chain particularly, this long single line of amino acids that are there. It's a single chain that forms the hinge region. Now, what do you understand by the term proteolysis? Good. Now, breaking down of proteins, how do you break them down? Enzymes. What are enzymes? Proteins again. Right? So you've got proteins that basically go down and cut down other proteins. Now, when you do that, there's something called again a steric hindrance. So there are areas of a protein that an enzyme can come easily to and cut, versus areas where it's difficult for the enzyme to reach and chop it off, right? So there are some parts that are exposed and can be cropped more easily versus others. So when you take papin, which is used for, how many of you like lahm? Oh, yeah, by default. So this is one question I have. Why don't all of you have high uric acid levels? Because you are just primarily meat eaters, right? Arabs, they can't do without lahm, right? So I would think you guys would have high levels of uric acid, which you don't, but anyway. So when you try to tenderize meat tenderizers usually have proteases, things that go and break down proteins to make it soft, and one of them is papin, which you primarily get from papaya. This is a weak protease, meaning it doesn't cut very strongly, and when it does, it actually ends up cutting at the hinge region. After this treatment with papin, when you try to crystallize the protein and you look at it, you get three different structures. You get two fragments that are identical, which are called fragment antigen binding, because these are the parts that are actually involved in antigen recognition. And one fragment that is left off is called FC, which is primarily fragment crystallizable, because it tends to crystallize very easily, and you can then do crystal studies to see the structure. Now, based on this, they understood the function of the hinge region. So the hinge region is a very important part of the antibody because it gives it flexibility. A single antibody has two antigen binding sites, right? That also means that antibodies by default try to target antigens that are repetitive in nature, therefore two would bind together, and it also means that these antigens that they go and bind to need not be spaced far away molecularly. So if something is spread out too far, they may not be able to reach it to begin with. Okay, do you get the point? Therefore, the hinge region actually gives it a little help. If the target antigen is too close, the molecule shifts its shape at the hinge region to bind to the target antigen closer. If they're a little too far off, it can always open up again. But again, the size of the antibody is a limitation. If you have a molecule which is far away here, it would not be able to bind to both simultaneously. It would only bind to one of them at one time. Okay? So the hinge region gives it all the flexibility to actually go and engage the target. Now, I remember one of you mentioning that the different kinds of antibodies is primarily you have different varieties based on the heavy chain and the glass chain, right? So, and that is very true. So, the different antibodies we had from IgG, IgM, IgD, A and E. So, the IgGs are called IgGs because of the constant structure of the heavy chain. Okay. Similarly, IgM now over here has got actually 5 globular domains instead of 4. And this one is an IgM, and again the heavy chain is of the mu type. Then you have IgD, again 4, IgA and IgE, again 5. So all of them have different constant parts of the heavy chain, and because of their different amino acid sequences, they can be highly glycosylated, meaning you can have a lot of glucose moieties being added to it, or actually monoglycosylated. So you can have poly or monoglycosylation depending on the kind of antibodies. Over here, IgG does not really get too many glucose moieties attached to it as compared to the IgD or the IgA in different parts of them. Yes, any questions till this point? So till now we're just going through the structure. Now these are proteins, right? And proteins should come from genetic material, DNA, right? So you have within the gene sequence within your genome sequence, you have the variable part, and we are talking about the heavy chain only, okay? You have a variable part that primarily goes and codes for the variable region of the heavy chain. And then you have constant parts. The constant regions, the sequence of them, how they are present in the genome, is that after the variable, you have the constant part of the mu, which is IgM, or the constant part of the delta. Okay. After that, you have constant gamma 3, constant gamma 1, alpha 1, gamma 2, gamma 4, epsilon, and the alpha 2. And if you go back one slide, you can see that IgG are further classified into 4 subtypes, IgG1 to IgG4. Similarly, IgA is again of 2 types, IgA1 and IgA2. And therefore, the gene sequences, the exons that kind of code for alpha 2 and the alpha 1. Okay? So this is how they're present in the genome. Is that clear? And in between, they've got intronic spaces. Now, we know how protein synthesis takes place, right? How does it go? The sequence. You've got the DNA, from which you get what? mRNA. And this guy has to go to the ribosomes, right? Exactly. But you will have tRNA coming in, bringing in the nucleotides. Okay? The mRNA acting as a template. And then the building blocks, amino acids being attached, and the protein being made. And if this is at the interface of the endoplasmic reticulum, you may have additional proteins in here, which are called as chaperones. You know what chaperones are? Yes. Exactly. Chaperones are additional proteins that help the chain to fold properly as it is being synthesized. Now, on the surface of the B cell, we're talking about a mature, naive B cell. You have IgM or IgD. These are called as B cell receptors primarily. And if you remember from yesterday when we studied, they have a small transmembrane domain that is hydrophobic, that helps us to anchor, and then very small cytoplasmic tail, and of course the Ig alpha and Ig beta are associated with them to help in signal transduction. Now, if you look at this structure over here, you can see that this part over here is the promoter region. Okay? So from the promoter, the first thing that is read is the exonic sequence of the variable chain, right? Followed that by constant sequence of the mu and the constant delta. Now, right after delta over here, not shown in the image, is where you have a stop codon sequence. So when the segment is read, it is read as V, C mu, C delta, and it stops. So the initial mRNA that will be generated will just stop at this particular point. After this, you have what you call as the mRNA, which is made for both IgM and IgD, and therefore at this point, it goes through alternative RNA splicing, meaning the RNA is cut between the mu and the delta, and either the delta is pasted to the V or the delta is removed and V continues with the C mu region. Okay. Now, this area, the point at which it's going to be cut, is where you have polyadenylation taking place. So, in case of an IgM expression, from the gene segment you will have the C mu and the C delta, all of it being converted into the mRNA. Here, polyadenylation will take place right after the C mu region. Okay, and these extra parts of segments that you're seeing here, these are the parts that code for its anchoring in the membrane. Okay. At this point, you will have other enzymes that will come and do alternative RNA splicing, and you will just have the final mRNA that is generated, which is that for only the C mu, the IgM heavy chain. Okay. Similarly, if you look at the other end, you will have polyadenylation taking place at the back of it, and the intervening segment, which is for the C mu, being removed, cleaved off, and the final mRNA that is made out of alternative RNA splicing will give rise to the IgD. So at any given time point in a resting B cell, the genome sequence only allows the formation of IgM or IgD. Okay, that's why these are the first antibodies that exist. And when people talk about IgM as the first antibody synthesized, it is because the genome sequence is already fixed for it. Now IgD is a non-secreted protein. It does not get secreted. It's not known to be secreted. Okay. In your bloods, however, you may have 0.02% of that antibody, but that is coming out as a result of the breakdown of the cells. You get the point? Just like you have baseline levels of ALT, AST, because the liver cells are dying off and releasing their enzymes, you have a similar amount of IgD over there, but that does not mean it is secreted. Okay? Sorry? When your cells die, what happens to the products? So they need to be phagocytosed. If they are not, they get released into their environment.
Why they are not?
Why they are not released?
Yes.
We don't know. Honestly, we don't know. There is no logical reason for why.
Doctor,
Okay. Yeah.
so the alternative splicing refers to IgM?
Both. It goes both ways. It is a randomized process. So your cells end up having IgD or IgM on their cell surface. A B cell ends up having both, and it's almost equivalent. Okay. There's no predisposition for one or the other.
But
Now-
at this point now, we don't have the same structure, right? The last three blocks, we don't have the same structure in the heavy chain, let's say.
Sorry.
Now the structure of
Yeah.
the heavy chain and the last part of the light chain are not the
Light
same.
chain is not being discussed at all right now.
Okay.
We're just talking about the heavy
part,
chain.
The blue part of it
Yeah.
and the purple part of it.
Yeah.
They're not the same structure. Different.
Same to what?
In the composition, let's say, or something.
So this is different. IgM is for IgM, and IgD is for IgD.
It's not only the last, it's not only the variable part. The whole thing is for IgM.
So the whole thing is for that. Okay? And we are just discussing right now how the constant part is being generated. We're not worried about the variable part that does the antigen recognition. So your different antibodies, the different classes, is based on the type of heavy chain they have. And what we're discussing right now is the gene sequencing, that the genome sequence that exists for different heavy chains, and therefore the different classes of the antibodies. Okay? Now, let's now review what happens when you have the light chain. The light chains are of two types. They are kappa light chains and lambda light chains. Okay? And again, in the light chain, they just have a constant region with another variable with a joining segment. So from this, you just end up having a variable part and one constant, either kappa or a lambda light chain. And in the heavy chain, you have a higher variability within the variable region. Now, is this part first clear to all of you? You understand how the heavy chain is assembled? Yeah. It's just the alternative RNA splicing. And this is in a resting B cell, a non-activated one.
So it slides in the after the IgM, the rest will go to IgD or
Yeah. Okay. Now, finishing with the heavy chain and the constant part of it, let's discuss a little more about the variable segment. How is that made? So, you recall we were discussing yesterday that you have around more than a billion different types of B cells, and each one of them having different kind of affinity for different kind of antigens, right? Now, since all your B cells end up having IgM or IgD at the surface, the only thing they can actually differ from each other is in their antigen binding site, the variable part, right? The variable part of the light chain and the variable part of the heavy chain should be different from one B cell to another B cell, right? because the rest of its structure, rest of the proteins are primarily the same. They don't differ from each other. They're again IgM or IgD, but it's the variable segments that vary. This part. Now, this is very important for you to understand antibody diversity and how they engage different kinds of antigens. So for the whole variable part, there are 3 gene segments that are available. And within those, there is something called as variable, diversity, and the joining segments. Together, when they combine, they give rise to one big segment that we call as the variable portion. Okay? Is this clear to you? Till here? Now, the variable, diversity, and the joining segments that are present are actually present for the heavy chain. Okay? For the light chain, you just have the V and the J segments. Now, the variable segments are approximately 1 to 80 different types. So V1, V2, V3, all the way up to V80, for example. Diversity range between 1 to 20, and joining are another 30 to 40. Okay? So the variability that comes in random joining of these 3 segments. The easiest example I can give you is if you have playing cards, okay? You've got spades, you've got hearts, you've got clubs, and you've got diamonds, right? So you get rid of the diamonds, okay? So you're left with only 3. The hearts, the spades, and the clubs, okay? Now you mix them up in different combinations, and you are supposed to have 3 only of each. 1 from hearts, 1 from spades, 1 from the clubs. So you can, on a lucky day, you can have 3 aces. On a bad day, you can get all 2s. Right? Yeah, I know how it works in your minds. Okay? But at the end of the day, you will end up having different combinations. Right? So similarly, your gene segments, the DNA, has around 80 of these, 20 to 40. And 1 is pulled out from each in a total randomized sequence. So you end up forming a VDJ over here, which is coding for the variable, and it may be V7, D2, J29. And another one, so this could be coding for this cell, and another sequence that may come in could be V19, J1, and D1 and J21. So these DNA sequences that are different end up coding for different kinds or different sequence, amino acid sequence of the variable part. Sorry?
Variable part of heavy chain.
Variable part of the heavy chain and then the variable part of the light chain.
So the point is like there is a whole set of variability within the heavy and then there's a whole set of variability within the
Exactly. And that further combines them to give rise to another different level of variability altogether. Right? And there's another point to it. The places where they're joined to each other, because this is DNA, right? A part of DNA is cut and pasted. Cut and pasted. So the part that comes in between that is actually joining, you have random nucleotide insertions. Okay? That further enhances the variability of it. Okay? You get the point? So, in the end, for the light chain, you have 2 gene segments, the V and the J, that give rise to the variability along with the constant light chain for the kappa or the lambda. And for the heavy chain, you have V, D and J that combine, first the DJ combines and then the V to the DJ, giving rise to the whole variable part over here. So this VDJ that you see is primarily coding for one variable segment of the heavy chain altogether, right? So till here we discussed how the gene segments code for the constant part, and those gene segments are fixed. What gene segments code for the variable part, and they are in the genome, they are fixed, but then the DNA is cut, spliced, pasted together to form truncated parts which are random and therefore different from each other. And then the variable and the light chains come together, giving rise to the overall variability that you see in the antibody. Now remember, within one cell, right, you have the antibody structure is one heavy and one light, and then another heavy and then another light chain. So these heavy chains and light chains actually have identical variable parts, because the gene coding for one heavy chain and the other heavy chain will have the same variable. Do you understand that part? Good.
Doctor, it's always
Yes, cerebral necrosis. Yes, sorry?
always VDG.
So for the heavy chain, it's always VDJ.
No, it can't change the.
No, the sequence remains as such. And we'll discuss it. So don't worry about it, but we'll discuss it later in detail when we do T-cell receptor rearrangement. That's when you really understand things better.
Sorry, Dr.
Okay.
The V that we have for the partner of heavy chain will be the same V for the light chain.
La. So one heavy chain and another heavy chain. Both have one gene segment coding for it. So the V for one will be the same for the V for the other, because it's a mirror. Okay?
But for the variable part, they will be different. And in such manner, the both variable parts will have the same V. Like this.
Exactly. Okay? Right. Now let's go to another part which we were talking about class switching. So class switching is primarily all about the heavy chain changing. Okay? Those who are practically dead in their brains.
Doctor,
Yeah.
you have constant light chain, correct?
Sahi?
How does it make the same way as constant variable chain?
Yes. But the constant light is fixed. Okay. Okay. Slow down. Let's go. Look, this segment is talking about the heavy chain. Okay? The heavy chain is made up of two regions we talked about before. Let's go a little back. Here we are, talking about life and how it evolves. So this is an antibody structure again. This is your heavy chain. Within the heavy chain, you have a constant part over here and a variable part. Within the light chain, you have a constant part and a variable part. The constant genes are fixed in the DNA. Also, the variable genes are fixed in the DNA. But the constant genes do not get reshuffled. They remain as they are. The variable genes get reshuffled, like we talked about, like the deck of cards. Okay? Now, going back to how all of that happens, is that in your DNA, the sequence is that when it is reading over here, this is the place where it's going to have a start codon and the promoter region. When it's going to read the DNA segment, it's going to start by reading a variable part, after which it will read, the reading frame will be that for the constant part of the mu, which is going to be IgM, and constant part of the delta, which is going to be IgD. And after that, there's a stop codon sequence and it stops. After this, in the DNA, you still have C gamma 3, which is IgG3, IgG1, IgA1, IgG2, IgG4, IgE, and IgA2. You still have those DNA sequences present for the constant chains for other types of antibodies, the class switch type of antibodies, which are present. But because there is a stop codon over here, that's it. That's what it does. So in a resting B cell, you have either IgM or IgD. Now, how is the heavy chain made? It's simply based on the reading frame, which will pick the variable part and the constant of the mu and delta, giving rise to the whole RNA. Polyadenylation chops it and gives rise to either an mRNA sequence that will give rise to the protein by a variable part with the IgM, O, the intervening segment is removed and the polyadenylation targets it for C delta. Okay, the polyadenylation takes place at the end of the chain, and this is relatively randomized equivalently, so you have almost equal numbers of IgD or you have equal numbers of IgM. Yeah.
Isn't the IgD should be longer because you got more terminally than the IgM?
Sorry?
So in IgD, you got a bit further than IgM, so the chain itself should be longer, not shorter.
This got nothing to do with the protein. We are still at the mRNA level. And we're talking about the exons that have been transcribed into the mRNA, and this is mRNA being cut. And mRNA post-cut being pasted together. Okay? So either you have the intervening segment of the mu removed and the variable joined to the delta constant mRNA.
But it should still be longer because the part of IgM is still there.
No, the IgM, that's what I'm saying. The IgM intervening segments is removed. You have a whole group of enzymes that come in the middle to cut it off. Okay? So again, repeating. After the variable part, you have sequences here that only read till the variable frame. And then the mu constant and the mu delta. Okay? And there are intervening segments which are intronic there, that are there where you can have enzymes which come and cut and remove and then paste the mRNA together. Okay? So for the IgM, it is easy because polyadenylation right at the end of the chain gets rid of the delta, and you have a RNA transcribed which gives from the variable and the constant region. For the IgD, you have the variable part and the segments in between of the C mu are removed. Okay? So this is how you end up forming either an IgM or an IgD. Now the question is, what about this variable part? This variable part. What codes for this variable sequence? Okay? And where do they come from? For the light chain, you have a single constant region, and for that variable part, you have two gene segments, a variable and a join. And between them, there are different varieties of them available in the DNA, and they are cut, pasted together by a process called somatic recombination. So starts with the germline DNA, the DNA is cut, and the VJ pasted together. Then you have the primary RNA transcript, which gets spliced to give rise to the mRNA, and the protein synthesized. So this variable part of the chain is actually coded in the DNA by a variable and a joining segment together. They together give rise to the variable part of the light chain. As for the heavy chain, you have the variable, diversity, and the joining segments. You have the diversity and the joining segments pasted together by first somatic recombination where the intervening DNA is removed. Then between the variable and the already joined diversity segment to the diversity and the joining, you again have intervening DNA removed till it gives rise to a VDJ sequence together. This all together primarily codes for the variable part of the heavy chain. Okay, yeah.
We have two copies of the genes, one for the mother and one for the father, so which one is used?
So if all of this fails to produce a protein on one, then it will go to the other one. Okay? So first, because this is DNA cutting and pasting, there is a probability that after the cutting, the pasting doesn't happen very well. Okay? And if it doesn't give rise to any recombination event, then it goes to the next gene. Okay?
Doctor,
Yeah.
is the constant part of the light chain the same in G and M and D?
So constant part of the light chains are only of two types, kappa and lambda, of all. Okay? So there is no variability in them per se. So IgG, IgM, IgA, IgD, they can have either a kappa light chain or a lambda light chain to it. That's it.
A specific sequence will have
Constant sequences are fixed for the light chains, kappa or lambda.
It's not like the process we said, the cutting of M, joining of M or D.
No, here joining of V and J takes place in the variable part of the light chain.
Yeah.
But the constant part is either kappa or lambda and fixed. Okay? It doesn't change. So in the end, you have what you call as a VDJ segment. Now the next part is class switching. Goodbye. We stopped. Hello.